Radiation therapy is most frequently delivered using linear accelerators. This protocol allows researchers to assess the biological effects of radiation on cells using a linear accelerator. The advantage of this technique is that by using a linear accelerator, researchers can study a wide range of clinically relevant doses and dose rates. Furthermore, this protocol can be used for all types of cancer cells.
This method can be performed using similar equipment to that shown here, for example, linear accelerators manufactured by other vendors. Individuals not experienced in performing dosimetric calculations using a linear accelerator may struggle to calculate the correct number of monitor units required to deliver the desired dose to the sample. They would benefit by seeking advice from an experienced medical physicist or dosimetrist. Delivering dose to the sample accurately using a linear accelerator requires careful placement.
Two days before scheduled irradiation, working in a culture hood, use a sterile 5 ml pipette to collect glioma stem-like cells from the culture plate into a 15 ml centrifuge tube. Centrifuge the cells at 200 times G for 3 minutes in a countertop centrifuge. Discard the supernatant and resuspend the cell pellet with 1 ml of trypsin EDTA. Incubate at room temperature for 5 minutes to digest the pellet and make a single-cell suspension in trypsin EDTA. Every 2 minutes during digestion, gently shake the bottom of the centrifuge tube to make sure the cells are thoroughly digested. Then add 3 ml of stem cell culture media to quench trypsin. Gently mix and centrifuge at 200 times G for 3 minutes in a countertop centrifuge.